Hello, and welcome to Talking General Practice, the podcast from GP Online. I'm Emma Bauer, the editor of GP Online. Today on the podcast, we're looking at the work of UK Biobank, a pioneering research resource that has the potential to transform our understanding and treatment of a wide range of diseases. I'm speaking to Professor Naomi Allen, who's a Professor of Epidemiology at the University of Oxford and UK Biobank's Chief Scientist. In this conversation, Naomi explains what UK Biobank is, how it was established, and the wealth of data it holds on its participants. She also discusses the crucial role of primary care data, how it will be incorporated into UK Biobank in the coming months, and what GPs and practices need to know about this. And we discuss some of the groundbreaking research that's already been conducted using UK Biobank data that could revolutionise patient care in general practice in the future.
Hi, I'm Pat Anderson, editor of MIMS Learning, and I'd like to invite you to join 2 free webinars on the topic of cancer diagnosis. First, on 23rd of April at 7pm, GP Dr. Sunita Kochhar will talk about diagnosis of skin cancer. And on 7th of May at 7pm, GP Dr. Pipin Singh will hold a case-based discussion with oncologist Dr. Pauline Leonard about how to achieve better outcomes for patients with cancer. Sign up now at mimslearning.co.uk.
I'm delighted to be joined on the podcast now by Professor Naomi Allen, who is the Chief Scientist for UK Biobank and a Professor of Epidemiology at the University of Oxford, to talk about UK Biobank, what it is, what it does, and how some of this work could impact on general practice. Thank you so much for coming on the podcast, Naomi.
Thank you for having me. It's lovely to be here.
Brilliant. So before we get into a bit more detail, can you just explain in the very broadest sense what UK Biobank is?
So UK Biobank was set up about 20 years ago now as a large-scale population-based cohort study in order to enable research into the potential causes of disease. And so we've recruited 500,000 volunteers across the UK between 2006 to 2010 in their midlife, between 40 and 70. And we've collected a whole range of data on them, and we've followed them up over time to find out what happens to their health. And we do that through linking to their NHS electronic healthcare records. And the beauty of UK Biobank and why it's so unique in the world is that we make this really fabulously valuable data set available to approved researchers worldwide. So the world's greatest minds can access these data and analyse the data to find the potential causes of disease and therefore help to find treatments and preventative measures for disease. So it's a database to enable health-related research that's in the public interest.
What exactly is your role with UK Biobank and how did you get involved with all of this work?
As Chief Scientist, my role is really to think about the scientific strategy of this amazing resource. So what will researchers around the world, what data do they need to really answer their killer research questions that otherwise they couldn't answer? So in addition to all of the data we have on lifestyle, genetics, and clinical health outcomes, should we be collecting data on circulating proteins, RNA sequencing? Should we be linking to environmental data? Should we be linking to data on pensions and benefits and all of these different types of questions? How can we enhance the resource to make it even more valuable to answer the most important research questions of the day? So my job is really to think what research questions are going to come up in the next sort of 3, 4, 5 years to enable the resource to collect the data to answer those questions.
So you mentioned your participants there. When you recruited them, they were all people in midlife, as you mentioned. Are they representative of the UK population? Who exactly are they and how did you get them all on board?
How we recruited these 500,000 participants is we identified about 22 different assessment centres around the UK, and they were centred in major cities, including cities where we had high ethnic minorities, so areas like Hounslow and Bradford and Birmingham and so on, to have as much variety in ethnic makeup, socio-demographic makeup as possible. And what we did was we sent postal invitations to everybody who is aged between 40 and 69 who lived within about a 25-kilometre radius of each of these 22 assessment centres. So we sent out about 10 million letters. And of those 10 million invitations, 500,000 people actually came along to an assessment centre, spent 2.5 hours with us and joined the study. Now, because our participants are volunteers, they're not representative of the general population. So on average, they tend to be a little bit healthier and a little bit wealthier than the general population at large. Having said that, because of the size of the study, we still have thousands of participants who live in very deprived areas. So that means that if you're doing an analysis, look at the association between deprivation and, say, let's say heart disease, you still have enough variability across that measure to enable any associations that you find to then be generalizable to the population at large. The only area where I say UK Biobank is perhaps not best place to do research if you're looking at differences in associations by ethnic group. So at the time of recruitment, which was the mid-2000s, 95% of our cohort are white, which was representative actually at the time of recruitment of Britain. Now about 11% of our population are ethnic minorities. So if you want to look at associations of, let's say, deprivation and heart disease across different ethnic groups, then you're going to rapidly run out of numbers to do that analysis in a robust way. So UK Biobank is wonderful for many things, but if you want to really look at differences in associations by ethnicity, then we don't really have the numbers to do that.
So you've got 500,000 participants. What data do you actually hold on them, and how do you keep that data up to date? How often do you get information from them?
So we've collected an awful lot of data on these 500,000 volunteers. So when they joined the study 15 odd years ago, they came along to a physical assessment centre in one of these city centres. They completed a touchscreen questionnaire about their lifestyle, physical activity, sleep, medical conditions, diet, and so on. Collected physical measures on them, ranging from height, weight, BMI, lung function, bone mineral density from the heel, blood pressure. They also provided us with a blood, urine, and for the last few thousand participants, a saliva sample. Recruitment ended in 2010. Since then, we've been continuing to collect data from our wonderful participants through sending out web-based questionnaires to our participants. So this is really to find out extra information about things that we didn't have time to look at in detail at the assessment visit. So do things like a 24-hour recall of their diet. Can they tell us a bit more about their occupational history? And to also collect information on health outcomes that are not well represented in medical records. So things like cognitive function, depression, other aspects of mental health, pain, ADHD and autism, and so on. In addition to that, about 10 years ago, we sent out wrist-worn devices, so like smartwatches, to 100,000 participants to collect objective measure of physical activity and sleep. We've also been undertaking an imaging study over the last 10 years, whereby we're bringing back 100,000 participants from the cohort to a dedicated imaging suite for across the country to have a 5-hour visit with us to undertake brain, heart, full body MRI scan, carotid ultrasound, full body DEXA scan, plus all of the measures that they took at baseline, which is why it takes so long, including biological samples, to enable researchers to look at how genetics and lifestyle factors influence internal physiology through the MRI scans and how that then relates to hard clinical outcomes. So it's the biggest study in the world that's got real multimodal data in terms of lifestyle, imaging, accelerometer. And from the baseline blood samples, we've obtained genetic information, whole exome, whole genome sequencing data, proteomics data on circulating levels of metabolites, biochemistry measures, telomere length. So you can see why people say UK Biobank is just a treasure trove of information. It's the largest, most detailed biomedical resource, and it's available to researchers worldwide, and thousands of publications, over 14,000 papers have now been published using UK Biobank data, and it's about 10 a day. It's really proven to be a real success in terms of collecting high quality standardized data on vast numbers of people and making this data available to researchers in a highly curated and confidential way.
Obviously, there's a possibility you might find something's wrong with people. Do you have processes for dealing with that in case the people spot anything?
We do have a process for dealing with. So it's worth saying that generally speaking, we don't give any feedback of any results to participants. This is what participants consented to when they joined the study. And there are many reasons for doing that. In the imaging study, however, if the radiographer happens to see, for example, a lesion in somebody's brain that they think may be troubling, they will then escalate that to the radiologist. If the radiologist agrees, then we will write to the participant and their GP to say, this is a research scan, it's not a clinical scan, but we have spotted something that we might think be serious. You might want to get it checked out. And about 2% of participants, we send a letter along those lines too. And in about half of those cases, it turns out to be nothing at all. And half of those cases, they'll obviously get it seen to. So that's been our process, and that's been working very well.
You've got all these samples. Are you storing those as well, so that basically as technology develops and things come along, you might be able to get them out of the deep freeze and test them for in a different way if some new development happens in the next decade or so?
Yeah, so our strategy actually, when we recruited all of the 500,000 participants, was we collected the blood samples, urine and saliva, but it's mainly the blood samples that are of high interest to researchers. We aliquot it into different types of sample like plasma, serum, buffy coat and so on. And then we just stick it in a freezer and we store it. And then we waited many years, actually, we didn't do anything with them until technology improves, until costs comes down, that actually it makes sense and is cost effective to do large scale, i.e. assays on every single person, so that the data is there for example, for the genomics. We waited many years until actually the price has sufficiently come down, the technology was there, so that you could genotype all 500,000 participants at one time in a highly standardized way, curate that data and then make it available to the research community. And we've done that for whole exome, for whole genome sequencing, biochemistry measures, and we're now doing that for circulating proteins as well. And of course, the beauty of doing it, having all of these measures available on everyone rather than a subset of people with, say, breast cancer and a subset of people without breast cancer is what was usually done, is that you've got that data for the whole cohort. So it enables vast numbers of research questions to be answered at the same time. And that's really been critical in the success of UK Biobank.
So you've got all these participants now that are taking part in UK Biobank. How do you keep them engaged and how do you decide what information you need from them?
So we send out an annual newsletter to all of our 500,000 participants, telling them what's happening with the resource, what extra data we're collecting. But we also have a participant advisory group. So this is about 20 participants, very diverse group, all sorts of expertise in different areas. We ask them advice about what we should be doing, what our strategy should be over the next few years. We get their feedback on certain types of new measures that we're thinking of introducing, to get their feedback and their input on data collection, information materials, what things should look like. And that has really helped us to engage much more effectively with all of our participants.
This is a closed cohort, isn't it? What does that mean and why is it a good thing that you're set up in that way?
We recruited our 500,000 volunteers between 2006 and 2010. And so we're not recruiting any more participants into the study. So that's what we mean when it's a closed cohort. We're just not open to more recruitment. We get asked lots of questions about, well, why don't you expand the cohort? Why don't you recruit the children and the grandchildren of the participants, which is a great idea. But actually, our participants, they're now average early 70s, age range between mid-50s to mid-80s now. We've probably got a relatively narrow window of opportunity to collect as much data as we can on these participants as they're entering older life. And I think we should be investing our resources on collecting as much data as we can on frailty, aging, cognition, social interactions, and so on our existing participants. From an epidemiological perspective, once all the participants have died, which should say be in the next 20, 25 years, that's when the resource becomes the most valuable, because you've got clinical endpoints on everybody, and the resource will be of use for research purposes for many decades after that as well. And you've also got the stored biological samples. We've used less than 10% of the samples collected at baseline. So this resource will continue to reap benefits many years after all the participants have died.
Is that why you kind of focused it on midlife to begin with?
Yes, we focused on midlife because they're old enough that you don't have to wait too long to have large numbers of heart disease, cancer, dementia, but they're young enough that most of them anyway haven't developed major disease yet. That was why we focused on the sweet spot of age 40 to 70.
How are you funded as an organisation, and how did UK Biobank come into being, as it were?
It really came into being through our core funders, which is the Wellcome and the Medical Research Council. So about 20 years ago, it was them who came up with the visionary idea, actually, of creating a really large-scale population-based cohort in the UK that was large enough and deep enough that it could really answer definitively questions about the determinants of common disease. It was their suggestion, actually, to make this data set available to researchers worldwide to maximize the value of the data. So the Wellcome and the Medical Research Council are our core funders, along with some other charities like Cancer Research UK, British Heart Foundation. Our model is that for researchers who would like to, for example, conduct an assay on the blood sample, for example, whole exome sequencing or whole genome sequencing or proteomics, then this has tended to be commercial research groups, namely big pharma and tech companies, who've come together to form a consortium to invest in UK Biobank to pay for those assays to be done, because these measurements, they cost tens of millions of pounds to do them in 500,000 participants. And so this model has worked really well because we make the data available to commercial companies at the same price and at the same terms and conditions as academic researchers. They will then often invest in UK Biobank by performing these otherwise very expensive assays on the samples. And then those data are made available to all researchers in a very equitable and transparent way. So that model is pretty unique to UK Biobank and has really been successful.
Are you very unique, or are there any other projects like this anywhere else that people might have heard of?
There are projects like UK Biobank for sure. So you've probably heard of our Future Health in the UK. So that's currently aiming to recruit 5 million British volunteers into a large-scale study where they collect a blood sample, they do pretty light touch measures of weight and height and blood pressure and cholesterol and so on. And then again, they follow participants' health over time through linkage to NHS records. There's a study in the States called All of Us. They're recruiting 1 million participants across the country with a focus on trying to get at least half of the cohort from ethnic minorities. And they've also done whole genome sequencing, lifestyle, physical measures, and linkage to medical records too. And those data are also now being made available to academic and commercial researchers. There's studies in China, there's studies in Singapore. And it is important that you have these types of general population bioresources available in different parts of the world, because a finding that you've got a genetic risk score associated with heart disease in the UK may not be applicable to Africa because of the different distributions of different genetic variants in those populations. So it's really important that you have these types of studies placed in strategic parts of the world.
One of the reasons why we're talking today, though, is because UK Biobank is going to have access to information from your participants' GP patient records. What do GP practices and GPs need to know about this? They don't have to actually do anything to make this happen, do they? But, as the data controllers, I'm sure they want to know what's going on with this data.
First off, if we were able to include primary care data into the resource, I don't think it's an understatement to say it would transform the type of research that you could do, because it means that the GPs are the first port of call for the vast majority of health-related issues, and diseases like arthritis, dementia, asthma, diabetes, which are largely diagnosed and managed in primary care, their numbers of cases will literally double if we had access to primary care data, because at the moment we only know that people who have those diseases through the hospital inpatient records. So if we have primary care, it enables researchers to find out what are the genetic and lifestyle determinants of these very common but debilitating conditions. And it will also allow detection of cases at an earlier stage. So this means researchers can then study the full spectrum of disease severity and bring new diagnostic tools and treatments closer. So that's the real added value of having primary care data is absolutely huge. So in October of last year, Wes Streeting, the Secretary of State for Health and Social Care, made an announcement that kind of paves the way for coded primary care data to be shared with consented cohorts. Previously, we've had to obtain every GP practice's approval to get the release of the primary care data to UK Biobank for participants who are in their practice. Now, given how overworked GPs are, how little they know about UK Biobank, and why should they, no matter what incentives we give, less than 20% of GPs would approve the release of the data. And because they're data controllers, they ultimately have liability for the data. So to be honest, I'm not surprised they didn't approve the release of the data, because it's much safer just not to do anything. But the announcement from Wes Streeting end of last year means that NHS England are now taking over the liability of the data. So this means that the data will flow from the GP system suppliers, TPP and EMIS, through to NHS England and then onwards to consented cohorts once NHS England have got all the necessary approvals and assurances in place for us to handle the data. And I think that centralised mechanism is really the only way it can work. That's what happened with COVID. We had access to primary care data for COVID research purposes only, and that was through emergency legislation from the government. It's exactly the same process as working here. GPs don't have to do anything. They don't have to press any buttons. There's no work involved for them. But we're really keen that GPs are aware and are informed about what's happening as possible, so that if they have patients coming to them who are UK Biobank participants, they know that their primary care data are being made available to UK Biobank for research purposes, of which participants have given their explicit consent.
They've basically all signed up to that as parcel of being part of this study, have they? Right?
Absolutely, yes. So at recruitment, all 500,000 participants gave explicit consent for UK Biobank to link to their health-related records.
And what information will you get? It's just the coded information, isn't it?
Absolutely right. It's just the coded data. So coded data on diagnoses, on prescriptions, on referrals, on lab values. It does not contain any free text, and it doesn't contain any attachments. So no letters from consultants back to the GP. So it is just the coded data. That's an important point. None of the free text is coming to UK Biobank, which will be a bit of a game changer in terms of the type of research and the number of cases of these common conditions that researchers will now have access to.
Is there any specific rules about who can use UK Biobank, or can anybody do it? And how does that process work if people want to get access to the data?
So we do have quite a stringent vetting procedure for who accesses the data for obvious reasons. In order to register as a researcher, what we call a bona fide researcher. So, you have to come from a research organization, have an institutional email address, you have to upload your CV, tell us what research you've done in the past. We will do quite detailed due diligence checks to make sure that anybody who wants to register to use the data is a bona fide researcher. And then when they want access to the data, they submit a relatively short online application form. They tell us about the research they want to do, what data they want to use, how they're going to disseminate the findings. And then we will, on the UK Biobank side, make sure that, for starters, is the research feasible? Can you do it in UK Biobank? Do we have the right data? And second, is it in accordance with participant wishes? So is it health-related research, and is it in the public interest? And if it is, then they can have access to the data on our secure research analysis platform. Access to the biological samples is obviously a higher bar because they're limited and depletable. So for researchers who want access to our samples, we need to make sure that the assay that they want to measure is validated, the data is going to be high quality, they're going to do it for the full cohort or at least a very large subset of the cohort. It uses a minimal amount of sample volume, and so on and so forth.
So how many people are currently using your data? I mean, you talked earlier about the number of studies that's been produced. It sounds like there's a phenomenal number of people actually making use of the data at the moment.
There's 20,000 registered researchers around the world. The top 3 users of the data are the US, the UK and China. There are about 4,000 active ongoing projects as we speak, and around 14,000 published papers over about 80 different countries around the world.
Some of the people listening to this may well have first heard about you during the pandemic. I think that's the first time I really came across UK Biobank, because you were very involved with research around COVID-19. As you mentioned earlier, that did actually make use of general practice patient records. Can you explain some of the projects you were working on during the pandemic and whether any of those are still ongoing?
We set up quite a few studies actually during the pandemic. So very early on, the government asked us to set up a surveillance study so we could monitor just the spread of COVID across the country by different age groups, across different regions, deprivation and so on, and look at really what were the main risk factors for having a positive test. We set up this surveillance study across the country, and we asked our participants, we recruited 20,000 participants, and we also recruited, for the first time, their adult children and grandchildren, because he wanted to look at COVID spread in the younger generations as well. From that data, we then showed that having a positive COVID-19 test was more common in the elderly, but it was also more common in ethnic minorities and in people who worked in the healthcare sector. Things that we all know and take for granted now, but it was quite an important study at the time. And we also did a study that looked at how long antibodies last following an infection. And so this required us to send kits to people's homes to take a blood sample and send that back in the post every month for 12 months. And then we did one at the 18 months time point to see how long do antibodies last over time, because we had no idea then, well, if you got the infection, how long are you likely to be protected for? And we found out that actually 90% of your antibodies are still there at 3 months, 79% are still there after a year, and over 70% are still there after 18 months. And then finally, we set up an imaging study whereby we kind of halfway through this imaging project anyway, where we're bringing back thousands of participants to have an MRI scan, when we thought, wouldn't it be great if we could invite some people who had already had a scan before COVID, invite some who've had COVID and some who haven't had COVID, and have a second scan. So we could determine the actual effect of SARS-CoV-2 infection on internal physiology and compare that directly between those who'd been infected with COVID and those who hadn't. And that research, there's no other research like that done in the world, where you've got a before and after MRI scan. And that research did show actually that those who've been infected with COVID did have changes in parts of volumes of their brain, particularly related to olfaction, and also slight reductions in cognitive function in certain domains once you've infected with COVID. And most of these COVID infections, they weren't serious, they weren't hospitalized, they were mild infections like most of us had. But it will be interesting to see what the longer term effects are on cognition. Has the brain recovered in terms of the volume of the olfactory region, for example? So it'll be interesting to do that again. But it was the only study in the world that we're aware of that was able to do that kind of research.
There must be lots of studies that are being published that potentially will impact on the way GPs either prescribe or diagnose or work out risk factors for people getting diseases.
Absolutely. I mean, it's hard to know where to stop, actually, but I think there are a couple of seminal examples which I think are worth highlighting. One is around the use of polygenic risk scores and identifying people at high risk of developing disease and how that might influence GP practice. So a polygenic risk score is where you look at all the genetic variants across your genome, each one of which may only add a small increase to your risk of developing disease, but when you add them together, may actually be sizable. So UK Biobank was the first study to really enable the development of this idea of polygenic risk scores because we've got genomic data for all 500,000 participants. And studies have been done where you developed a polygenic risk score for heart disease. And that pilot study was implemented in the NHS where they looked at the added value of adding this polygenic risk score for heart disease into the Q risk algorithm. Over above age, sex, blood pressure, cholesterol, all the things you plug into the Q risk score. Does adding in your genetic risk of heart disease, how does that influence the 10-year risk of developing heart disease? And what this pilot study showed was that in about 1 in 8 patients, the GP would change the clinical management of the patient based on the addition of the polygenic risk score. So, for example, their risk will go from medium to high, and so they might prescribe preventative statins, as an example. So that's a really good way to demonstrate how the genetic data could be included into an already established algorithm like Q-risk score to help risk stratification of high-risk patients. Obviously, it's not implemented yet, but we've kind of made the proof of principle that through this pilot study that actually polygenic risk scores in some cases can have some clinical utility.
Are there also studies going on that's sort of looking at helping us understand risk factors and preventative strategies for certain conditions as well? Is that part of the use the data will have?
Oh, yeah. So one of the main reasons UK Biobank was set up is to enable research, epidemiological research into what are the genetic and lifestyle determinants of disease. And then once you know what the potential causes of disease are, then obviously that can lead to downstream new treatments and preventative measures. So there's lots of research being done on dietary risk factors, physical activity, air pollution, obviously coupled with genetics, how these may interact to cause disease, which then in time will absolutely lead to new treatments and new preventative measures. One other area which is only now gaining real traction is the area around the importance of measuring circulating proteins. So I think this could be even more important than knowing about our genetics. So we've done a pilot study where we've made data on about 54,000 participants. We made available data on about 3,000 circulating proteins. And there's lots of research being published now showing that your protein profile is highly predictive of many diseases. So, for example, a handful of proteins can predict your risk of developing dementia up to 10 years prior to a clinical diagnosis. can predict your risk of developing a range of cancers 7 years before an individual is clinically diagnosed with a disease. You can say, well, wouldn't it be great if you can have a simple blood test that could measure your protein profile and be able to stratify groups of individuals who are likely to be at high risk of developing disease based on their altered protein profile, that could then go for either earlier screening or more preventative measures or what have you. So it's still early days, but I think this area could potentially really change the way we identify early disease and how we identify people at high risk of developing disease in the future. You know, obviously your genetic profile doesn't change. You're born with it. Whereas your protein profile changes based on what you eat, how much you exercise, whether you smoke, and also early pathology. You know, many years before you're diagnosed with Parkinson's, dementia, cancer, heart disease, that early pathology will change your protein levels in some cases. And so you can use that protein profiles as early warning markers of disease before you've got clinical symptoms even, or as a high risk factor for disease. And so you can then add preventative strategies or screening measures to those who are high risk. So personalized medicine is not as sexy as it was 5 years ago. And people talk more about tailored treatments and tailored therapies. But I think this could be the real road through which we could travel down. And over the next 5-10 years, I really wouldn't be surprised if at our annual health check, there's some kind of measurement of protein levels coupled perhaps with genetics.
As you said, the participants in your study, they're getting old now, and this is obviously where you're going to get people with multiple long-term conditions potentially interacting with each other. Are you hopeful that the data is going to be better at giving a picture of how we deal with this situation we're going to be in, where we're all living for longer and most of us are living for longer with a number of conditions that may interact with each other?
One of the key strengths of the primary care data is the fact that you've got such comprehensive information about multimorbidities. Because the hospital data, you know, it's very good at coding what you came into hospital with, and sometimes it codes what other conditions you've also got, but it doesn't code everything. And is only at the time of admission, whereas the GP data goes back many years and you've got really comprehensive information about those multimorbidities. And now already research is being done on multimorbidities in relation to cancer risk and how that affects cancer survival. And it's the same with dementia and other, non-communicable diseases. Is multimorbidities becoming a huge problem? The more research we can do on the impact of multimorbidity on both incidence of new disease, survival, and the burden of disease, the more that will enable GPs and the NHS more broadly to help manage those conditions.
And just before we finish, if there's any GPs who are involved in research, there's lots of GPs who are very involved in research. If there's anyone listening to this who've not made use of UK Biobank data and think they might like to try, or there's something they think they're working on, how do they go about doing that? Do they just need to come to your website and fill in that form you were talking about earlier?
All the information's on the website. So we're just updating our website, actually. So in April, we'll have a very streamlined, very pretty website, and hopefully it'll be a lot easier for people who are interested in using UK Biobank. And I hope some GPs will be, particularly if you can collaborate with data scientists, epidemiologists. We're always looking for clinicians for their input into research. Have a look on the website. All the information's there. Register to use the resource, and it's very easy to submit an application.
Yeah, that's brilliant. Thank you, Naomi, for coming on the podcast today. Thank you.
Thank you very much. It's been great.
Well, that's it for this week. Thanks so much again to Naomi, and thank you for listening. I'll be back next week for our regular news review, so please do join me then. In the meantime, don't forget you can keep up to date with all the latest news affecting general practice and access a wealth of other resources on our website at GPonline.com.